think I'll record because, as you know, it's snowing out there. And you guys know why you guys are here and my Monday classes won't be here? Is because chancellors don't come in on Sundays. Sorry. And that's recorded, so some boss is going to fire me because I wisecrack. But of course, no chancellor is actually going to listen to a teacher's lecture either. So I guess I'm pretty safe. Oh dear, I'm cracking myself up. So all right. So 4 levels of protein structure. This is a pretty complicated concept, but it's also a concept that some of you have probably had before. So raise your hand if you've had this, if you faced this concept in another class. 1, 2, 3, 4, 4 of you, right? So did you guys see those hands raised? Those of you who didn't raise your hands, put those hands in the air. I've already learned about 4 levels of protein structure. Several of you, yeah? Those of you who didn't raise your hands, there they are. Okay, so here is the flashcard basics. There's 4 levels of protein structure. There's primary, secondary, tertiary, and quaternary. The names make sense, don't they? Yeah? And these levels are they are layered on top of each other. They all exist in most proteins simultaneously, okay? But we talk about them one by one because they help us understand how proteins are built. Is that clear? You guys get me? Okay, so what is primary? Well, it's the amino acid sequence of the protein. Again, this might, if you understand what this is saying, good on you. If you don't, I'm going to explain all these to make this make sense to you, hopefully. Secondary, backbone interactions. Tertiary, side chain interactions. Quaternary, multiple polypeptides interactions. As we said before, as we said last week, proteins are complicated. They are so complicated that computers not only can't solve them now, can't hope to solve them in the future. They are impossibly complex. Okay. And that's because of these levels of structure. Let's look at them individually. At the most basic level, a protein is a polymer of amino acids in the same way that starch, glycogen, and cellulose are polymers of glucose. Does that make sense? Take 1 glucose, you've got glucose. Take 2 glucoses, link them together, you've got maltose. Take 1,000, and you've got starch, or glycogen, or cellulose, depending on how you link them together. Yeah. So take a bunch of amino acids. Again, instead of taking one monomer and linking it together in a few different ways, we're taking any one of 20 different monomers and linking them together one by one by one. So here is a protein, and I can't remember what protein this is, but I think it might be insulin. You guys know what insulin is, yeah? It's a pretty important protein. So how do you make insulin? Well, start with lyse, which is lysine, then add val, which is valine, and then add phe, which is phenylalanine, and then gly, which is glycine, and then add your pirate amino acid, arginine, and then so on and so on and so on, yeah? Sorry, how many times have I cracked that joke with this class? Once. No, this is now twice, yeah? Okay. So I've only got 10 or 20 more times that I can crack that joke before I have to sort of give it up, right? So there are 129 amino acids in this protein, yeah? So it's a recipe with 129 steps, and those steps are recorded in the DNA for the gene for insulin. Is that clear? Guys with me? All right. So, but insulin is much more than just a ribbon of amino acids. We've got a picture of insulin at the beginning of this. I'll backtrack so we can look at that. Do you see it? Insulin is a bunch of squiggles. You see that? And those squiggles, right? We have a squiggle here. There's spirals here and so on, and there's some other yellow stuff. Yeah? These squiggles hold the thing together. So once we make our initial sequence of amino acids, right? Comes from linking together the individual amino acids one by one with peptide bonds. We talked about that last week, yeah? Okay. So once those amino acids are linked together, remember there are functional groups that form those amino acids, yeah? There is a double bonded oxygen here. There is a NH group here, an amino group. Well, as it turns out, NH groups and double bonded oxygens can do what when they come close to each other? These little dotted lines, what are they? Anyone recognize what these little dotted lines are across here in this spiral and across here in this zigzag? Those are hydrogen bonds, yeah? Hydrogen bonds, where do you guys, again, you should be familiar with those already coming into the class. Where do we usually see hydrogen bonds when we talk about what? Water, right? H2O forms hydrogen bonds, yeah? But it's not the only thing that can form hydrogen bonds. Do you guys remember hydrogen bonds? Shall I take 2 minutes to, let me just briefly review hydrogen bonds. And again, you guys should already know this. And it is foundational. But let me just briefly remind you. Here's H2O. You recognize the shape, a little bit bent, yeah? Here's another H2O. If you learned about electronegativity, or if you didn't, you know that the H's are a little bit positive, just slightly positive, and the oxygen is slightly negative, right? Well, negatives and positives, what do? They attract each other, right? So this positive and this negative have a little magnetic attraction in the same way that there's a little magnetic attraction between your magnets and your refrigerator, yeah? They can, and that attraction is weak, you can pull it apart, but it is there, yeah? Okay, likewise though, it's not just water molecules that can form hydrogen bonds. Anything where you have oxygen bound to hydrogen or nitrogen bound to hydrogen, you can form hydrogen bonds, yeah? And it happens within the backbones of amino acids, right? All along the backbones of amino acids, every single amino acid has an amino group and a carboxyl group. Every carboxyl group has a double bonded oxygen. Every amino group is an amine group, yeah? So hydrogen bonds can happen between here and here and here, all the way down the whole length of the polypeptide chain, the basic structure of the protein, yeah? Okay, so there's two main, what we call motifs. Do you guys know what a motif is? A motif is a sort of a, is an artistic convention, yeah? Can someone think of an example? The term usually comes from art. How many people know the term motif? So a motif is kind of like anyone listen to popular music? Yes, well that's good. I have one. Did you guys know I had one student in the class that listens to popular music?
I think everybody does.
Raise your hand if you listen to popular music. Okay, within popular music there are genres, yeah? Raise your hand if you wish to share a genre that you are familiar with in popular music. Country. Have you guys heard of country music? I've heard of country music, yeah? So, country music has thematic motifs and fashion motifs, yeah? Like any genre of music, you will see country music performers dress a little differently and sing about different songs or sing different songs about different kinds of topics, right? And use different kinds of melodies. Give me a different genre of music besides country. Rock. All right. So rock and roll versus country, the performers in rock and roll also dress differently, yeah? So what would country, so if you saw a cowboy hat with a particular kind of brim, right, that would be like a kind of motif for country music, yeah? Kind of like a hallmark. Does that make sense? If you saw a leather jacket with silver snaps all over it, you might think that was more of a rock and roll thing. Is that a fair assessment? Okay, so if you saw long hair or something, you might think that was metal or some genre like that. Is that clear? So these are familiar motifs in the same way a cowboy hat is familiar to country music, whereas a leather jacket with silver snaps on it is familiar to rock and roll. Is that clear? So these two motifs are the alpha helix and the beta sheet, okay? And what we'll see is those things crop up when you look at the structure of proteins all the time. Here's some examples. Can you see the alpha helices? Which one has alpha helices? The one on the left. These two spirals are a alpha helix of secondary structure, yeah? Which ones have a beta sheet? Looks like both of them. This one has a green sheet that's flat. This one, the sheet has been wound into a barrel. Is that clear? Right? So those alpha helices and beta sheets are beginning another level of structure, yeah? And the next level of structure is tertiary structure, right? When we take this two-dimensional shape and we fold it into a three-dimensional barrel, we're getting another level of structure made out of the second level of structure, yeah? At all proteins are made out of a single ribbon of amino acids, right? You can see this ribbon in these pictures, can't you? Where does it start? Looks like it starts here with the letter N. It zips along, goes this way, then it goes that way, then it goes up, then it zips around, and then it starts doing the helix, and then it goes up again, then it goes back down, and then it comes back here and along, and then it starts doing the helix again, and then it ends here, right? So it's linear, yeah? It is a single ribbon that is coiled up in a particular special way. You guys got it? But then that coil forms a three-dimensional shape, yeah? So that coil, those ribbons, the way that protein initially folds, it folds on itself again. You understand? And that folding again is tertiary structure, and the thing that makes that happen is the side groups. How many different side groups? 20 different side groups. That's what makes the 20 different amino acids all different. Yeah? All amino acids are pretty much the same across the backbone. All 20 amino acids are significantly different on their side chains, right? Some are acidic, some are basic, right? With my little French slide that I like doing every semester, right? Some of them are polar and can do hydrogen bonds, right? Here are hydrogen bonds on the left here, yeah? Some of them are acidic and basic, and the acidic ones form a negative ion, the basic ones form a positive ion, and the negative and positive ions attract each other, and an ionic bond can form between those two, yeah? Likewise, nonpolar side chains like each other too. Is that clear? You guys get me here? Okay. So I think this picture illustrates the concept well. This is a light bulb filament. Raise your hand if you like to stare at light bulb filaments. All right, so maybe you guys have better things to do with your time than staring at light bulb filaments. I mean, they're fascinating, aren't they? So behold, the humble light bulb filament. How many spirals is it made out of? I hope you see at least 2 spirals. Can someone explain that? Look at it again. You see the spirals? 2 spirals? Raise your hand if you see it. Got 2 people. Everybody else, try to see it, right? Within this picture, I believe, is a big piece of understanding what the difference is between secondary and tertiary structure is, okay? So, where's the spiral? Is this the spiral? That's the spiral? But that's one of them. It itself is made out of a spiral. See that? So, take your metal wire, spiral it up, coil it up, yeah? And then do what? Take your coiled wire and coil up your coils. You understand? So, this coil here, what's that like? That's the secondary structure. And then this big coil, what's that? The tertiary structure. Does that make sense? Right? It is folds. Your folded thing can be folded. Yeah? Your coiled thing can be coiled. Does that make sense? You guys get that? All right. So, tertiary structure, again, we take our coiled thing and we take our sheeted thing and we fold it around to make a barrel. Yeah? The barrel is the tertiary structure. The sheet itself is the secondary structure. Yeah? Does that make sense? All right. So, as it turns out, tertiary structure has a lot of interactions. Do you know how we talked about hydrogen bonds in water and so on? Do you know how we break hydrogen bonds? Raise your hand if you know how to break a hydrogen bond. Yes. Hydrolysis. What do you think? It's not hydrolysis. How do we break? So hydrolysis breaks a covalent bond that holds monomers together. Yeah? High temperature is the answer, right? When water molecules have all the hydrogen bonds they can possibly form, what do we have? There's a hydrogen bond between all 4 of these. Right? We have water. What kind of water? Solid water. What do we call solid water? Blankety blanket snow or ice, yeah? That is where we have a crystal, right? Ice is a crystal, and it's a crystal of water molecules where every single possible hydrogen bond that can form is formed in that water molecule, yeah? And that's why it's rigid. That's why it's solid, yeah? And when all of the hydrogen bonds are broken, right, then there's nothing holding these water molecules together anymore. Now what do we have? When all of the hydrogen bonds are broken, we have gaseous water. What do we call that? Steam, yeah? That's water vapor, right? Where all the hydrogen bonds have broken. Does that make sense? So what about liquid water? As it turns out, liquid water is a state of water where the average number of hydrogen bonds is somewhere in the 1 to 3 range, right? every water molecule should be able to form 4. And when all of your water molecules do form 4 hydrogen bonds, you have a crystal of ice, right? And you can see how that crystal forms, yeah? When all of them are broken, the water molecules are free, yeah? And they float around freely, and they are a gas, yeah? As water molecules cool, they condense, right? And one water molecule will bump into another, and they'll orient themselves to form a hydrogen bond, and then they'll accumulate in the same way that you ever roll up snow to make a really big snowball, right? So they sort of stick to each other in the same way, and then you, instead of getting a really big snowball, you get like a water droplet in a cloud, yeah? And you get a cloud formation or something. All right, so the take-home message, though, is that hydrogen bonds can be broken with heat, yeah? Not just in water, but any hydrogen bond is broken with heat, yeah? So what happens when we heat up proteins? Their structure gets changed. They get denatured, yeah? So do you see this? There's a hydrogen bond holding this part together, yeah? If we heat up the protein, this hydrogen bond gets broken, right? What happens to the protein? Well, this part and this part are no longer held together. They can flop around. Does that make sense? What happens when we heat up egg whites? Aren't they proteins? Yeah, it's albumin, which is a particular kind of protein that's common to a lot of vertebrates, yeah? So the shape of the protein changes, yeah? The protein changes permanently, yeah? Reverted the shape of the protein? I would love to see that article. How interesting. So generally not reversible unless you have a very, very clever scientist, yeah? And how would the scientist know how to reverse the... Well, he would study the shape of the protein, understand the shape of the protein. Egg white albumin is a protein that certainly has been crystallized and must be understood, right? Again, without being able to have a look at the shape of that protein, that guy would have a lot less of a chance of being able to figure out how to rewind the damage done by cooking it. Fascinating. Okay. Guys get me though? Proteins are fragile. They are precision machines, yeah? And precision machines can be broken, just like a microscope, yeah? Or anything, yeah? That's the more sophisticated the machinery, the more fragile and delicate it tends to become, yeah? So there's a lot of ways to mess up the shape of a protein. By changing pH, by making things more acidic or more basic, we can also denature proteins. Anyone ever eat ceviche? Raise your hand if you like to eat ceviche. It's yummy. Did you just say the OMG, yeah? Because it's so good. How do you make ceviche?
You
Cook raw fish in lemon juice. So, what would you say about cook? She said the word cook. What does that usually mean? Heating. Do you heat it? You don't. It's cold. And yet the acids, you take a bunch of lemon juice and squirt it onto a raw fish, and it changes the fish. The proteins change with acid in ways that are similar to the way proteins are changed with heat. Both heat and change in pH will mess up the tertiary structure of your protein and denature the protein. Has anyone ever put lemon juice on a salmon before they put it on the grill? You see it kind of burn the surface of the salmon, right? It burns it in the same way that grilling burns it, right? You have your salmon before you put it on the grill. What does it look like? It's sort of a peach color, yeah? And it's flexible and flimsy, right? You throw it on the grill, what happens? It becomes whiter, it becomes flakier. It becomes stiffer, yeah? You take lemon juice, you squirt it over salmon, the same thing happens. in a little longer time, but a similar thing happens. It becomes whiter, becomes flakier, becomes stiffer, in the same way that grilling the fish does, yeah? Likewise with a hamburger, right? You have a hamburger, it's red and gooey, you throw it on the grill, the proteins change, yeah? They go from being red and gooey to being gray and stiffer, yeah? You agree? Right? The proteins change, and they change permanently. You put your grilled burger in the fridge, it doesn't revert back to the original shape of the burger. Is that clear? And you put your fried egg in the fridge, it doesn't go back to the liquid consistency of a fried egg unless you have some chemist who does something. A chemist who does that has done something very clever, yeah? All of you would agree. Okay, great. So lastly, quaternary structure. Again, first, primary structure, the sequence of amino acids. Secondary structure, the initial interactions of backbones to form hydrogen bonds to make helices or beta sheets. Tertiary structure, the folding of helices or beta sheets using the interactions of the side chains to make complex structures, three-dimensional structures. And then lastly, quaternary structure is where we take multiple polypeptides, multiple ribbons of amino acids, and link them together. So a good example of that is hemoglobin. Are you guys familiar with hemoglobin? Each of these, there's 4 pieces to hemoglobin, 1, 2, 3, 4, and each of them, 2 are similar, are one shape, they're the orange ones. The other 2 are pink, they're a different shape. This is alpha and beta hemoglobin subunits. It takes 4 subunits, 2 of each of 2 different kinds of subunits to make a single hemoglobin molecule. Is that clear? Likewise, to make collagen, it takes 3 individual ribbons of the same kind of polypeptide to make a ribbon of collagen. Okay, so mess up the amino acids in a protein, you mess up the whole thing. You change the primary structure of hemoglobin, right? Here's regular hemoglobin. Here's hemoglobin with one mistake, right? Glutamine, amino acid number 6, turns into valine, and as a result, what do you get? Well, the original shape of the protein looks like this. The changed protein looks like this, right? Red blood cells full of hemoglobin looking normally, and then full of misshapen hemoglobin, suddenly your red blood cell is changed, yeah? What do we call this? Sickle cell anemia is caused by a mutation in a single piece of DNA that changes one amino acid in one protein that has far-reaching effects, right? Have you guys heard the expression, for want of a nail, the shoe was lost? For want of a shoe, the horse was lost. For want of a horse, the rider was lost. For want of a rider, the message was lost. For want of the message, the battle was lost. For want of the battle, the kingdom was lost. Yeah? Say it again. You heard that somewhere. How many people have heard that saying? Right? That saying is, have you guys heard of what's called the butterfly effect? Yeah, so where occasionally, sometimes when you change one thing, that thing changes another thing, which changes another thing, which changes another thing, and the consequences of one small change can be significant down the road. Does that make sense? Right? In the same way that losing that nail out of the horse's shoe had big consequences for whatever kingdom was talked about. Does that make sense? Likewise, you change one amino acid and you change the protein. You change the protein, you change the shape of the cell. You change the shape of the cell, you change how that cell flows in your body. You change how that cell flows in your body, and you can have health effects when you're trying to exercise, yeah? And those you have That's sickle cell anemia, yeah? All right, so here's summary of protein structure. This is from your textbook. Here's the primary sequence. Here's your two kinds of secondary structure. Those secondary structures form tertiary structures. Tertiary structures stick together to form quaternary structure, which forms your overall protein. Got it? All right. All right, more than one polypeptide, what do you have? More than one polypeptide is quaternary structure, right? Here's one polypeptide, 2, 3, 4 polypeptides stuck together. That's quaternary structure. As it turns out, not every single protein has quaternary structure. All proteins have primary, secondary, and tertiary structure. Okay? Most proteins have quaternary, but some don't. All right, so much for proteins. Should we? Yes.